So I am Angela Page. I am Director of Strategy and Engagement for GA4GH. And for this next session, we've asked 3 different initiatives: the Human Pangenome Reference Consortium, the Public Health Alliance for Genomic Epidemiology, or PHAGE, and Seneca, to present on how they are currently engaging with GA4GH and where there may be opportunities for future collaboration with the work streams. So each initiative will have 10 minutes to present or so, and then we'll open up for a pretty lengthy Q&A for each talk. And we really hope that you'll all join in and ask some meaty questions about how we can drive these collaborations going forward. So to kick things off, we have Heather Lawson presenting on behalf of HPRC. Heather is an assistant professor of genetics at Washington University in St. Louis, Missouri. Her background is in molecular anthropology and quantitative genetics, and her lab focuses on gene by environment interactions and parent of origin effects. She recently joined the Human Pangenome Reference Consortium to assist in outreach efforts and to collaborate with the GA4GH in the formation of an international Human Pangenome Project. So thanks, Heather. Take it away.
Thank you. Yeah, and thank you for the opportunity to present an update on the Human Pangenome Reference Consortium and also to highlight ongoing efforts that align with GA4GH workstream goals. Next slide, please. So we can all appreciate that the human reference genome is the foundational open access resource of modern human genetics and genomics, having set the bar for data sharing and providing a centralized coordinate system for reporting and comparing results across studies. Next slide. Yet, even after 20 years from the first release, the human reference is incomplete. Because of the focus on euchromatic regions, we're missing regions that have been difficult to assemble because of their repetitive and highly polymorphic nature. Next. And further, the current reference is a mosaic of it's a mosaic assembly comprised of data from over 20 individuals, with a single individual actually representing around 65% of the sequence. So this linear assembly, it doesn't represent a real genome. And because it doesn't adequately represent global diversity, it can create reference biases, and this can affect variant discovery, disease association studies, as well as the accuracy of genetic analyses. Next. So our motivation for the Human Pangenome Reference Consortium is to actually improve representation of sequence diversity in the human population by sequencing 350 diverse human genomes and creating a comprehensive map of human genome variation. Additionally, we are aiming to provide a new reference data structure that will hopefully foster a new ecosystem of pangenome tools. And components of the HPRC include population representation and sampling, multi-center sequencing technology and production, reproducible workflows to reach finished telomere to telomere assemblies and foster variant discovery, open data sharing, cloud-based data management, human pangenome representations, and then of course, outreach and education to support implementation of this new reference. And next. So as such, the HPRC functions through working groups, and these working groups are focused on population sampling and representation, technology production, phased and finished telomere to telomere assemblies, data management and production protocols. And all of this is embedded in an interdisciplinary ethics working group and oversight committee. And through these working groups that we envision utilizing GA4GH foundational and technical work streams. For example, in the technical and assembly arenas, we're currently using VCF, BAM, and CRAM files in alignment and variant calls. And then we're also actively developing pan-genome pipelines that can emit VCFs to represent variations. Next. So how do we utilize GA4GH work streams going forward in this project? And I think ongoing work that's going to interest leaders and members of the work streams include developing graph construction standards, graph evaluation and quality control, as well as annotation, variant calling, and proof of principle for common applications. And I know recently the Genome Knowledge Standards workstream has presented their variation representation standard to the HPRC to facilitate future work that's aimed at extending this current standard to graph-based representations. And then we're also seeking global partnerships to increase sample diversity, to share data, as well as to coordinate on standards that we're using and that we're developing. Next. So I'm going to walk through some ways that we're developing sequence assembly and production standards. So assembly methods were evaluated in HPRC through a pilot method or a pilot benchmark study of a single individual who's a well-characterized sample from the Genome in a Bottle Consortium. So multiple long read and short read technologies, as well as computational methods, were evaluated to develop a combination of platforms as well as an automated pipeline that provides the most complete and accurate genome graph. And it was determined that trio approaches using parental sequence data to sort haplotypes of the offspring's long read data actually gave the most complete assemblies of each haplotype with the least structural errors. Next. So in the first phase of data production, which produced the first 30 genomes, the HPRC took advantage of work that was already completed by the 1000 Genomes Consortium. And there were three main principles that governed our initial sample selection. The first was to cover genetic and biogeographic diversity. The second was the availability of low passage cell lines for each of the samples. And then third, the availability of trios, so parent and offspring data. Next. And data production began in earnest across multiple institutions using multiple technologies. So the biorepository for the cell lines is at the Coriell Institute. PacBio production centers included Rockefeller University, Washington University, where I'm located, the University of Washington, and then the production center for the Hi-C data was UC Santa Cruz. Next. So I know this is a bit of a busy slide, but this graphic really illustrates our ultimate goal of a human pangenome reference through the efforts of many of the different working groups. And it also, I think, highlights areas that align really well with GA4GH technical work streams. So what I'm really going to do is just sort of walk through the alphabet here and describe our pipeline or general pipeline. So in A, we have this linear reference assembly as a backdrop, and then variants are depicted as new edges in the pangenome reference. And what this is going to allow is for a backwards compatible coordinate system, which we hope is actually going to encourage the use of the pangenome reference. In B, we're showing that haplotypes are inferred from genetic variation, and taking that to C, we're showing that this variation is characterized by iterating through each individual and then creating this genomic graph structure. And this is only going to improve as new genomes are added. D is a cartoon illustrating that genomic data is organized into an edge-based sequence variation map. And in E, we're just really saying that the RGFA file format maintains this graph structure, and it does so in a disk-efficient manner. Moving over to F, we're showing that samples can be genotyped by direct mapping against this graph. And in G, haplotypes are inferred by tracing a path through the graph and connecting sequences at the edges. And then ultimately in H, we would have this reference that catalogs genomic variation, and having this graph structure is going to allow for population scale analysis. Next, please. Thank you. So all initial sequencing data are currently publicly available, at least for the 30 samples that we initiated the project with, as well as some additional samples from other projects. And these data can be freely downloaded from the Amazon Web Services or analyzed with the Amazon Web Services Cloud. The data is also available for analysis within the Anvil Cloud platform, and this is the NHGRI's genomic lab space that provides a cloud environment for analysis of large genomic data sets and supports multiple globally used analysis tools. And of note, Anvil uses a number of GA4-GH standards in several places. Next. Thank you. So I want to finish up by going back to our population representation and sampling. So our goal is to create a resource that best serves humanity. And one thing we know is that the 1,000 genome data itself alone is insufficient to fully represent genomic diversity within the human population. Next. Is there another slide? I seem to be missing. Oh, it faded in. Okay. Okay, so what I want to do is actually end by introducing the Human Pangenome Project. So we were recently awarded a supplement, and I've joined the consortium to help with these efforts. We hope to achieve eventually an inclusive pangenome reference resource with global support that can improve clinical genomics and biomedical research, as well as provide opportunities for training, outreach, the sharing of resources, standards, and workflows, the ultimate goal being to form an international alliance of genomic partners and provide accessible, high-quality genomes representing the diversity of the human population, and these genomes will be represented as a pangenome reference. And towards these efforts, we've already begun some conversations with GA4-GH members, as well as with members of GA4-GH driver projects. And I think as the Human Pangenome Project move forward, there's going to be very important needs for employing GA4-GH work streams from several different arenas. And also as we're starting to launch the project and make plans for it, I think it's a natural fit for discussions with the regulatory and ethics work stream. And I welcome further discussions as we move forward. Next. And so just to sum up, I want to acknowledge a number of people, a number of institutions. Unfortunately, not all the people involved are listed here, but I want to point out especially Karen Miga at UC Santa Cruz and Ting Wang at WashU's Genome Institute for really helping me sort of get my bearings as I just recently have joined this project. And then I guess I will welcome any questions you may have. Thank you.
Thank you so much, Heather. That was great, and really exciting that you guys have managed to kick off this new international effort. So we have questions coming in on Slido, and it looks like there are also some in the chat.
Oh,
so I'll just go ahead and read them for you. So what will HG38 and chromosome 13 become when the genome graph is ready?
Yeah, so this is the current human reference, as well as the recent launch of the telomere to telomere genome. And what they're going to become is actually paths in the graph once it is ready. So we're going to have this backwards compatible coordinate system, but they will themselves also become graph paths.
Okay, great, thank you. So I'm going to read this question from the chat next. David Glazer says, I liked your A to F slide of multiple steps on the way to fully pan genome. Do you have target dates for 1, publishing a single graph that's good enough for many projects to lean on, including weighted edges to reflect frequency, and 2, having easy to use tools for new individual genomes to be genotyped as just a path through the graph?
Oh goodness. So yes, there are plans to publish. I know that currently we're working on presenting sort of the first pipeline through that benchmark study that I discussed, as well as initial results from the currently available sequenced genomes. I believe we are aiming towards an early 2022 release of the description of the HG002 pipeline. That would be the benchmark pilot study that I discussed. As far as your second question about tools, I know a lot of the technologies have not been published, but we're working on it. And so I think there's going to be kind of a paper drop next year. At least that's what we're aiming for. But in the meantime, the data are available to play with and explore, and we would welcome any and all comments, suggestions.
Great.
Yeah.
All right, next question, why 350 individuals?
Yeah, so this was actually decided, I mean, long before I joined the project and as it was just being planned out, but this was, I guess, in theory, the number that could capture all variants at a frequency that would be greater than 1%.
Interesting, okay, thank you. Next question, what is the sex ratio of these 350 individuals?
Yeah, so that's a great question. And it's actually approximately equal, but to have adequate coverage of the sex chromosomes, we're actually going to be supplementing with extra Ys, Y chromosomes.
Got it, okay, thank you. Final question coming in for now, what are plans to engage the broader community of genetics research?
Yeah, so this is really an open and ongoing discussion. Currently, our plans are to make everything that we're doing as transparent as possible. Obviously, outreach would consist of workshops at various scientific meetings, presentations to let people aware that this reference exists, that these data exist, and what our ultimate goals are. Invitation for the community to actually give feedback and be involved in the process as we're going along. And then specifically, I think we want to involve clinicians and clinical scientists. And this is going to mean maybe showing some concrete examples of the information that can be gained by such a diverse and comprehensive resource, which with some concrete examples, and then also maybe even amongst local populations that want to be involved in this resource, how we are able to address some specific needs as well as general population needs. So that's an ongoing discussion, and I welcome any thoughts, comments, or suggestions. We do have social media where we try to be as transparent to as broad an audience, maybe even reaching beyond scientists as well.
Okay, great. So there is one more question, but we are at time. So I hope that you could maybe answer that in the chat and then we can move on. Is that okay?
Oh, sure, happily.
When will the initial pangenome file formats be ready for the project be agreed? So I'll let you take that to the chat. And
okay,
then,
thank you.
Thank you. Our next presentation comes from Duncan McCannell, Emma Griffiths, and Danny Park, who will be presenting on phage. And I will go ahead and introduce each of you now, and then we can just dive into all the presentations at once. So Duncan McCannell is Chief Science Officer for the U.S. Center of Disease Control Office for Advanced Molecular Detection, where he helps coordinate the implementation and support of pathogen genomics, bioinformatics, high-performance computing, and other innovative laboratory technologies across the CDC's 4 infectious disease centers. Duncan played a key role in establishing the Public Health Alliance for Genomic Epidemiology, or PHAGE, and now co-chairs its Training and Workforce Development Working Group. Emma Griffiths is a research associate in the Center for Infectious Disease Genomics and One Health at Simon Fraser University in Vancouver, Canada. She is also the chair of the Data Structures Working Group within PHAGE, and her work focuses on developing and implementing data standards and tools to better enable analyses and the interpretation of pathogen genomics data sets. And finally, Danny Park is the senior group leader for viral computational genomics at the Broad Institute. Along with Pardee Sabeti and Bronwyn McInnes, he co-leads viral sequencing and analysis efforts at the Broad, including epidemiological analyses of viral outbreaks such as Ebola, Zika, Lassa fever, SARS-CoV-2, and unidentified viral fevers. Since 2014, Danny has led computational analyses and methods development in the group and has directed the implementation of computational pipelines on DNA Nexus and Terra for use by partners in H3 Africa and US Public Health. And Danny co-leads the PHAGE Infrastructure Working Group with Peter van Huytzen. And so it looks like we're going to kick off with Duncan and then just go through each of your talks from there.
Good morning. Thank you, Angela. Can everyone hear me?
Yeah, we can hear you great.
Excellent. Fantastic. So completely different topic. And thank you so much for the organizers for inviting us here to present today. Yeah, we would like to introduce or talk a little bit about the mission of the Public Health Alliance for Genomic Epidemiology. Now, it really should be no surprise to many in the audience that applications of microbial genomics in public health have lagged behind other elements and other sectors of the bioeconomy. Part of the reason for that is scale. You know, the types of applications that most public health labs, most public health entities need to apply genomics to are fairly large. The samples sometimes are inaccessible. And for many reasons, you know, these technologies just weren't implemented at the same speed with the same rigor that you saw in human genomics and other applications. The past 2 years, though, has really demonstrated the explosive potential of these approaches. Over 4 million sequences, SARS-CoV-2 consensus sequences have been submitted to public repositories. Many environmental and clinical metagenomes have been included along with those, and often with paired clinical samples. And so really pathogen genomics, public health pathogen genomics is coming of age. Back in 2018, one of the things that many of us in the community recognized was that sequencing was rapidly no longer the bottleneck in applying these technologies. It was becoming much more straightforward to roll out sequencing platforms and to train groups on how to sequence pathogens for different applications. Where we were starting to run into challenges was in bioinformatics and some of the data standards and data processing approaches. So in March 2018, we convened a small group of a couple dozen public health scientists from all over the world, thought leaders in microbial genomics, to really walk through the problem and to identify a strategy going forward. At the outset, we were trying to see whether we could improve the openness and reproducibility of microbial bioinformatics. And what we rapidly came to a consensus on, though, was the need for some sort of consistent standards organization or standards effort, or at least a venue to start having these conversations around how to implement these approaches at large scale. One of our first stops was to speak to GA4GH leadership to see whether they, really taking this organization as a template on what we were hoping to accomplish on the microbial side, to explore whether this was possible under the GA4GH umbrella. And I think we realized pretty quickly that this was a completely separate set of tasks, but very closely aligned in a lot of areas. So with that, we launched at the end of 2018 an organization that we call PHAGE. At the outset, it does have a lot of very similar organizational components to it. We have a number of work groups now. In the introduction, we mentioned several, including the data structures and workforce development working groups. But these have already started to have important conversations and start to develop products and consensus around some important issues, including SARS-CoV-2 sequencing and antimicrobial resistance analysis. My goal here today is to introduce the overall organization. I'm going to hand it off to Emma and to Danny to talk specifically about some of the key objectives and the efforts that have gone on over the past few years. Yes, I think I'll just hand off to Emma at that point. Or Emma, you on? Yes.
Yep. Great. Thanks, Duncan. So thanks for inviting us. Thank you very much. It's a great pleasure to be here to listen and to learn from everyone today. So in the data structures work group, one of our top priorities is the development, promotion, and implementation of data standards in public health settings to help increase the interoperability, reproducibility, and auditability of pathogen genomics. And one of the most common activities in public health pathogen genomic analyses is the detection of genes and mutations that correlate with different phenotypes, which can be often a lot simpler in microbes than it is in human genomics. Now, often there are a number of different tools that can be used for answering the same kinds of questions, and those tools can vary in terms of their scope of function, as well as the data structures used to encode their inputs and outputs, making results sometimes difficult to integrate as well as interpret, which then can complicate decision-making and communication about those results in service-based environments where information needs to be shared, integrated, and used very quickly. Because of this, one of our top priorities in the data structures working group was to develop a specification that could standardize fields used to structure gene and mutation detection and could prescribe the formats of values that go into those fields. The idea was to make the core of the specification agnostic to particular bugs or different use cases, but to enable modules to be able to be added on to customize the specification for different needs. We also wanted to create the specification with language that was familiar and made sense to public health practitioners, but we also wanted to make sure that all of the fields were also annotated with ontologies to help improve interoperability across domains. Next slide, please, Danny. So our first application of the specification was for the detection of antimicrobial resistance, which is, as you probably know, a massive problem around the world. And it's very serious because in many cases, antimicrobial agents are our last line of defense against all kinds of life-threatening infections. Now, genomics is being increasingly used to routinely identify and monitor the presence of resistance genes and also resistant bacteria in different settings. And for this use case, we augmented the core specification with additional AMR-specific fields, and we also created some tooling in the form of different parsers and also an interactive report to help put the standard into practice. We've also worked with health partners around the world, we're involved in different surveillance programs like the PulseNet Latin America and the Caribbean, as well as the WHO's Pan American Health Organization. And so we've been able to deploy the spec and its tools in a number of different pilot projects. Now, we know that there's been a lot of work done by GA4GH in similar efforts, particularly with respect to the VCF standard. And so we would really like to put our heads as well as our standards together to make sure that we're on the same page, and that so that we at PHAGE can also benefit from all of the wisdom of GA4GH. Next slide, please, Danny. As Duncan already mentioned, another project that we have been very busy developing is that of a harmonized contextual data or metadata specification for SARS-CoV-2 genomic surveillance. Now, most existing standards and repository requirements are largely based on capturing free text, which I'm sure you've experienced that makes the data pretty messy pretty quickly. Now, the specification covers a wide range of different data types, and you can see some listed in the slide here. And it has been implemented, the specification's been implemented in different sequencing initiatives and also by the INSDC as a biosample checklist. So whether users are future-proofing their own data for reuse down the road, or whether they're sharing with trusted partners or with public repositories, we're hoping that the spec enables the streamlining of analyses and different processes. Now, we are thinking that this work could probably overlap quite a bit with the PhenoPacket standard, so it would be great to work together with GA4GH to see how we can harmonize there for bringing public health data together with clinical data. And also, it would be great to explore whether it would be possible to create some sort of beacons for public health. So I think there's a lot of ways that our work could be aligning, and I'm very excited about possible projects that we could do together in the future. So now over to Danny.
Thanks, Emma. As Duncan mentioned, a lot of what we've also been trying to do in PHAGE is just harmonize and standardize ways that people are even approaching compute in the first place. Partly, the goal has been to really lift the long tail of smaller labs that can participate in data generation and consumption in the public health ecosystem. And part of the goal there is really to make it easier for a lot of places to stand up compute ability and to bring in tools in a faster way. I'm borrowing here a diagram or a concept that I've seen used before in GA4GH, or at least data biosphere circles, where just kind of thinking, it's been helpful to think about different types of users that interact with the compute platform. But I think part of what's been the interesting challenge in the public health context is that historically a lot of these actually, not for any particularly good reasons, but like actually are all the same people. And you know, even in a world where we have a kind of better separation between those who make the tools and those who use them, I think there's a lot more overlap here than we often deal with elsewhere. And so it's kind of led to, I guess, our starting point a couple of years ago is just like a huge ecosystem of just different ways to approach it, not just different pipelines, but just different languages, or if you're even using a standardized workflow manager, different kind of ways that infrastructure looks, a whole bunch of different kind of legacy, for lack of a better word, infrastructure setups that kind of have to be supported. And I'm not really kind of going into details here, but I think a lot of the work in the PHAGE infrastructure and the pipelines group has been trying to figure out how to shift this into a way that looks a bit more GA4GH in the future. I mean, these types of kind of pictures shown here are the types of places that we'd like to get to, but I think our big questions, almost philosophical questions that we wrestle with are there ways that we have to start saying that there are certain types of compute patterns that can't work in an interoperable ecosystem? Which types of languages or approaches play best, or being able to make those types of recommendations. So that's one of the things that we're currently running in. And I'll just touch on this really briefly because I don't want to go into the nitty-gritty details here. But for some of you know, we've been engaged with folks, especially in the Drury work stream, for a little while now, and I think this is going to pick up as we go into the next year or so, or next season of PHAGE. Without going into all the motivations, there's just like a need for figuring out better ways of facilitating low friction but protected genomic data sharing. And there's a lot of tools and things that exist in the GA4GH ecosystem that we would love to utilize and make use of and harmonize with. But I think I'll just end with this kind of general larger observation or kind of inference as we start to try to adapt a lot of GA4GH tools to public health applications, is that there are a lot of priorities and needs that are actually quite similar between working with microbial pathogen genomes and the different issues there, and a lot of what GA4GH usually tries to solve for. But there are some differences that make things a little, so for example, even just in the duo situation, a lot of the codes there, the goal is to make sure that use is compatible with patient consent and patient wishes. And we actually tend not to think about patient, like consent is not really a thing that comes into play when a lot of the data that's generated is generated under a mandate of public health. So there's other kind of interests and issues to kind of think about there, but it's interesting as we try to adapt the GA4GH tools to a different context. And so I think this is just kind of ending on the note that more broadly speaking, like Duncan said, there was a recognition that many of the things that we're trying to do in phage are highly aligned with what GA4GH is trying to do, but the application is just subtly different enough where we have found it useful to be able to drill in from a different angle. So I'll end with that. We really just wanted to put out an update of the types of things we were working on, kind of tease at some of the technical details of some of the bits and pieces that we're engaged with. We are already talking with some of you, but for those of you we aren't and you're interested, please contact us. Our info is here. And thank you so much.
Awesome, thank you all. We already have a number of questions coming in on Slido. And I think maybe we could group a few of them. So what are the next steps to facilitate collaborations between phage and GA4GH? And we have people asking which work streams are most relevant to your activities and whether there are common projects, work streams, projects that would benefit both the human and pathogen use cases of the two organizations.
Maybe I'll start at that first. I think those first two are related, and it came on the very last slide. We already have conversations with Dury and Cloud at the moment. There's a number of things that overlap too. The H3 Africa Driver Project has a number of folks who engage in phage as well. But part of our goal here is if you are not part of our engagements already and are interested, please do reach out.
And I think from the data structures work group perspective, we could really do with sitting down with some experts from GA4GH and having them walk us through some of their standards. I think that would really speed up our sort of absorption and integration process. So hopefully we'll be able to reach out and set some of those meetings up.
Yes, that sounds great. Someone has asked if there's scope for phage to attend some cloud sessions and share use cases. So it sounds like you have a first step right there. And I do think it makes sense, to take a look at the agenda and see which, for this week's meeting, to see which ones you might start those conversations on. What steps are being taken / can be taken to implement standards in public health settings?
So maybe I'll start off with this one. I think that what's really key is to be engaging public health practitioners in kind of all of the different stages. So right at the tools and standards development stage through, working out what our implementation strategy is going to be. It's one thing for us to come up with an idea and then try it out, but it's another thing to have gotten, to understand what the needs are and how systems work. So I think engagement right from the start is really key. Practically speaking, we also have a number of phage subgrants that we just rolled out earlier this year. So that was an attempt to put our standards into practice and implement those in public health settings, and in a way that we could learn and hopefully benefit those users. We're still largely engaging with academic labs that are working with public health labs. So we still have to do some work in better engaging those public health spaces and public health practitioners, but we are that's very much on our to-do list.
Great. Okay. Well, I think we've gotten to all the questions that have come in through Slido, and I don't see any others anywhere else. I know there's lots of places for people to ask questions. So if anything else does come up, please go ahead and put it in the chat, and then these folks can answer it by typing. And so we will move on now to our final presentation from Thomas Keene, who will present on Seneca, the common infrastructure for national cohorts in Europe, Canada, and Africa. Thomas is team leader for the European Genome Phenome Archive, EGA, and the European Variation Archive, EVA, at EMBL EBI, where he's responsible for strategic planning for both of those resources. His research interests are in using genomic technologies to understand biological processes with a particular focus on rodent models for human disease. And as you heard earlier, Thomas is also co-lead of the large-scale genomics workstream. He is co-lead of the Data and Interoperability and Connectivity Working Group of the International 100,000 Cohorts Consortium and coordinator of the Seneca Project, which he will now present on. Thank you, Thomas.
Hi there. Hi, everyone. It's good to talk to you again. I'm talking to you in a different context this time. So as Angela said, I'm going to tell you about the Common Infrastructure for National Cohorts in Europe, Canada, and Africa project, the Silica Project. This is a project that's joint funded by the European Commission and the CIHR in Canada. And it really is focusing really on how we try and deploy GA4GH standards to enable human cohort interoperability across a variety of different cohorts that I'll tell you about in a few minutes. Next slide. All right, so I guess just to give you a little bit of context, I mean, I think many of us know this and are aware of human cohorts, and certainly the emergence of many longitudinal disease-specific cohorts over the last many decades, actually. And we've got all this rich phenotyping, lifestyle histories. We've got various biobanks. We've now got national healthcare initiatives becoming more interested in making their data available, for research, secondary research purposes, and of course then expanding into generating huge amounts of genomic data. And I guess if you try to map out the cohorts, so there's a map on the right hand side of this slide here, which was produced by the International 100K Cohorts Consortium a couple of years ago, and really was trying to map out the different types of cohorts that are out there, the sort of ones that are coming from national healthcare, some of the big cohorts coming from research, and really looking at where are the largest cohorts, the one typically over sort of 100,000 participants. And you'll see that it is truly global. I mean, there are cohorts across all of the continents. And what we're going to see, of course, is healthcare is going to produce, we're already seeing it, vast amounts of genomic data that's potentially can be used in conjunction with the data to really make new discoveries. There are some cross-cohort interoperability initiatives emerging. I mentioned the IHCC, of course. The other one is the International Common Disease Alliance, ICDA, and then we have the various biobanking initiatives. BBMRI ERIC is one of those in Europe, and there are others out there that are, I suppose, working perhaps in particular diseases. Next slide. Okay, so what is the Seneca Project? I mean, essentially what we're trying to do is to enable population scale genomic and biomolecular data to be made available and accessible across international borders. And of course, we want to accelerate research and improve health of individuals across those continents. This is a huge challenge, and Seneca is not going to solve all of this. But I think in the 4 years that this project is running, we'll try to move things forward with the cohorts that we're working with. If you do the next slide, I'll just tell you about the cohorts. So some of the key features, I mean, I guess you could say the project is population scale. We're working with 10 cohorts with approximately 1.4 million participants. The cohorts they're not disease specific. They're not longitudinal. They're a mixture of all types of different cohorts. What we really were going for when we were designing the project was really to try and ensure that we had a sort of representation of cohorts across the three continents, and that was our real kind of driving sort of goal when we were establishing the project. Next slide. I think one of the fundamental sort of assumptions of the project is that we were really trying to move the federated analysis paradigm forward. And this was, again, given that we have this variety of different human cohorts across these continents, and we have a set of use cases, or research use cases and clinical use cases that research, typical research types of analysis that you might want to do, things like GWAS and EQTL analysis and some clinical applications. We could just go and do it in the traditional sort of way of accessing the cohorts, downloading the data and doing it all locally, but we really want to make this project a driver for the federated analysis paradigm. So there are a lot of parallels to the FAST project that we've heard about earlier. So you'll see there are many of the standards that are being deployed by the cloud workstream. We are also implementing those in the Seneca project. We're working with things like the Nextflow pipeline to get these standards sort of implemented at various different cohort locations, and then we can build the various federated applications across these. Next one. Yeah, I think, what the model that we're going with for the Seneca project is that you're not going to hear of the Seneca cohort. You're going to hear maybe of Seneca project being mentioned in different places. What we're trying to do is support current sort of infrastructure that exists, say, in particular, the Elixir initiative in Europe, the CANDIG initiative in Canada, and the H3 Africa BioNet project. Really support those existing infrastructures and initiatives to integrate and deploy and develop the GA4GH standards and deploy them across their infrastructure. So I guess we're not trying to build a specific Seneca system, but we want to actually move things forward in terms of deploying and implementing the GA4GH standards in these existing international sort of initiatives. Next one. And just to go through sort of what are the key sort of challenges. Again, I mean, these should be quite obvious to this audience. I mean, you can almost map each one of these challenges into work streams for GA4GH, but we were sort of laying these out in terms of what it means for human cohorts. So federated data discovery, federated cohort discovery, that's clearly a huge challenge. How do you find the cohorts of interest if you, for a particular analysis that you want to carry out, or a particular type of research that you want to carry out? And clearly, to be able to do that, you need to have things like harmonized cohort level metadata, which was our challenge 3. The whole business of data cohort access and this authentication authorization / passports in the sort of GA4GH language. That whole business is a major challenge, and I think we've made some significant progress in that area. We have a set of, we want, from our research and clinical use cases, and we really want to kind of move forward with the federated analysis paradigm and get that sort of working in the cohorts that we're working with, and certainly show how it can be generalized into much larger sort of cross cohort initiatives. And then, of course, we're working across 3 continents, so there is a huge LC challenge there and a huge LC framework. So there's some really quite interesting work happening in terms of mapping across the different LC, mapping across the different regulations that exist, perhaps in Canada and Europe and in Africa, and trying to see how they are compatible or incompatible with each other in terms of actually getting access to the cohorts for different types of research. Next slide. All right, so I'll just briefly go through the Seneca structure. I guess initially we start off with a set of cohorts. If you just do the next slide, there's a table here that kind of gives a listing of the cohorts we're working with. You can see here that there is, as I said, there's a whole mixture of different longitudinal cohorts, disease-specific cohorts, healthy cohorts, population controls, with all sorts of different data types. I mean, when you start to work with human cohorts, you really find that it's just a sea of different diversity and different types of cohorts that were built for different reasons with different data available. It would be great if they all were uniform and had the exact same data types. You can see on the right-hand side here, it's quite a sort of patchwork. But what we're trying to do then is map our research and clinical use cases and applications into which of the cohorts that are suitable for those use cases. So that's one of the processes we've been doing over the last year. Yeah, there's a variety of different sizes. You can see that they're based in different countries, which really makes the project quite fun, because we truly are trying to do sort of a cross-cohort interoperability between this variety of heterogeneous cohorts. Next slide. So I guess I'll just briefly go through some of the overall design sort of, and just briefly talk about the work packages. I think our work package 2 is all around the AAI and how do you get access to the cohorts. And I think most of the work for this, the project has been up and running since early 2019. So we're kind of coming into the sort of final 18 months of the project. And most of the work for our AAI work package was completed in the first, mostly completed in the first 2 years of the project. And I think, you know, one of the significant achievements for our AAI work package is that we've got the Elixir AAI talking to the Candig AAI and the Elixir AAI connected to the EGA, and all of this is done with the GA4GH passport. So we've really contributed to one of the sort of first implementations of the GA4GH passports, and getting these different cohort sort of hosting infrastructures to be able to talk to each other using the passports, so that users can identify themselves. So, you know, I think that has been one of the really great achievements from the RAAI work package. Next one. I guess one really significant area is this whole cohort level metadata, data dictionary mapping, trying to map the fields. I mean, the cohorts are collected at different times using different ontologies, if they use an ontology at all, or different control vocabularies, trying to get cross mappings of those cohort metadatas and fields and the data dictionaries is a huge challenge. And we're seeing this certainly in the IHCC project, that, you know, when you start to scale up to look across many cohorts, if you're working with one cohort, that's great. You know, it's feasible because you're just working with one cohort trying to understand their fields. But when you're working with multiple cohorts, trying to cross map between the cohorts and trying to map the different metadata fields is a real challenge. So this work has been led by Melanie Cartone and Fiona Brinkman, and I think one of the outputs we have from this is this Gecko metadata model, which has been, you know, we've had a really successful collaboration with the IHCC to roll this same approach out with some of the IHCC cohorts, building up to the launch of the IHCC cohort atlas, which you can see a screenshot for here. And I think this approach, it seems to scale quite well, and certainly I think we've got maybe, you know, going beyond Seneca and incorporating the IHCC, and then we're talking to some of the other cohort initiatives, some other disease-specific cohort initiatives, to see if we can continue to scale this Gecko metadata model from cross-mapping the fields in the cohorts. And all of the, you know, once this work is done, all of this is made publicly available, and this will live like long beyond the project. The output of these cross mappings, because that's really valuable work that's actually quite tedious and challenging to carry out. Next one. I guess once you've done that sort of cohort level metadata mapping, I mean, really that is almost a requirement for doing your federated data discovery, your cohort discovery by phenotype, genotype, data use perhaps. And I think the Seneca project has been a real driver for the development of the Beacon version 2 standard. And, you know, we've brought many of the use cases to the Beacon version 2, and we've also brought, you know, many of the sort of the cohorts that we want to be able to do these federated queries, discovery queries across. And yeah, next slide. Yeah, and all of this is building up towards the sort of latter half of the project, is to focus on our federated research and clinical applications. So, you know, we're talking about trying to do federated GWAS, trying to do federated EQTL analysis, trying to do, you know, use in our clinical applications, use the knowledge from the different cohorts to inform sort of potentially clinical decisions, or provide evidence to clinicians that we're working with. And I guess this is in our work package 4, and I guess this is really, well, 4 and 5, and this is really sort of backloaded once the kind of work of the cross-cohort metadata harmonization is done, the discovery portals are up in place, and the AAI is working, that we can really now focus on getting these applications. So we're kind of coming into the same kind of sphere, the FAST sort of project, trying to get sort of the end-to-end applications, federated applications up and running, and get those demos in place. Next slide. Yeah, and then this is, so it's just talking about, again, some of our federated clinical applications, and again, that's really kind of a highly active area of the project in this kind of last sort of 18 months of the Seneca project. Next slide. And of course, we're supported then by a variety of different work packages around our dissemination and training, and our LC work package is really highly active because there are some quite significant LC challenges in the project. And of course, we've got the project coordination and management led by Leslie Glass. Next slide. Yeah, so I guess if I was to looking back at when we were designing the project, if I was to think of like what were the kind of unexpected challenges that we had, I mean, one of the we realized, you know, maybe 6 months into the project, is that we have all these technical work packages that really want to get on with their demos and really want to start showing, you know, starting to get on with their development and showing, you know, building their demonstrators and be able to show those. But of course, trying to get access to human cohorts can take quite a bit of time. So we realized quite early on that we needed to have a set of synthetic cohorts that would be available for these kind of demo work and development and testing, etc. And that's something that we have spent quite a significant amount of time in building these synthetic cohorts. So essentially what these would be is essentially human cohort data that's unencumbered by pre-authorization and access processes. But of course, they will be loaded into all the standard sort of access processes, but they're not going to have to do all the kind of you know, data access committee sort of processes, and they can be openly shared amongst the project participants. We want all of the synthetic cohorts that we've developed to be available through the exact same discovery and data access methods as the real cohort data, so you can really build sort of realistic demonstrators. And the metadata that should be representative of the particular cohort that's being modeled, you know, as in using the same data dictionary fields, and maybe trying to at least somewhat mimic the range and distribution of values, of course, without having, you know, having the risk of any kind of exposure of the individual level data. We now have a set of synthetic cohorts. We've given these kind of generic names: Europe UK1, Europe CH SIB, and Africa H3F Africa Bionet version 1, Canada Child version 1. These are our first wave of synthetic cohorts. We're now working, and typically what these would have is some sort of genomic data, some sort of genetic data, and also some sort of cohort level metadata that is realistically modeled on the real cohort. We're trying to build a next generation set of these cohorts where we start to add in, looking towards more multi-omics. So we're starting to add in, you know, expression data and other types of omic assays to be able to make these a bit more advanced for our research and clinical use cases. Next slide. I guess the real focus for the project as we come into as we're coming to sort of the latter half of the project, is really to build these end-to-end federated applications. And this is, you know, really where you take one of our applications, maybe the polygenic risk score calculation, maybe the federated joint genotyping workflow that we're trying, federated workflow that we're trying to implement, and then getting it to use the discovery mechanisms, using the AI processes for multiple cohorts, ideally across the different continents. And then trying to either, you know, bring the analysis to the data using the various cloud workstream interfaces, and then trying to put all of the result together basically to produce the overall results. So it's really getting into the quite sort of fun part of the project, I guess, where we really start to see the kind of demonstrators and of these different research and clinical applications coming together. And I guess this is really where the kind of we start to hopefully see the real sort of results of the project coming together. Next slide. I think probably the most advanced at the moment of our research use cases is well, maybe it's the most straightforward, is the federated joint genotyping. So there is a video that describes this, how this works. I think this work was led by Kirill Tuskanov at EBI, and I think he's now got a quite good demonstrator up and running. And I think we've been using the Nextflow pipeline or workflow management system as the kind of backbone that kind of supports how this one has been implemented. And probably I think the other research and clinical application, well, certainly the research applications will probably also use the Nextflow workflow management system to support the federated application. Next slide.
Sorry, Thomas, not to rush you, but we have just a few more minutes for questions.
There you go, I'm done. Here's some links to the project. Here's a picture of the project participants from the pre-pandemic times when we had our one and only in-person meeting, and hopefully we'll have another one before the end of the project. And yeah, there's links to get information on the project. And yeah, and thanks to our funders, SCIHR and the EC, for funding the project. Happy to take any questions. Awesome,
thank you. Sorry about that. All right, so we have a few questions in here from Slido. Catherine had asked how Seneca and IHCC intersect, but then she said you answered her question, so I don't know if there's anything else you wanted to add there.
Yeah, it's kind of interesting. I guess we just happened to be on the same sort of timeline, I guess, and we realized that a lot of the work that we had been doing in the Seneca, although Seneca is a smaller number of cohorts, it's the exact same challenges that the IHCC really want to start addressing. So we really were able to transfer some of the work that we were doing in the Seneca project directly into some of the early IHCC work, in particular this harmonization and the IHCC Atlas, which we launched last year.
Okay, great, thank you. And Catherine seems like she really enjoyed your talk. She's also asked, you're already linked in with GA4GH, but if Seneca was external to GA4GH, what would be the best way for you to interact with us, example via FAST?
Yeah. I mean, I guess we're in the same space as FAST, I guess. The challenge for us is really that because we're working with these set of cohorts, and we're really trying to get the cohorts to implement things like the AAI and the discovery. So what we're trying to do is essentially bring the GA4GH standards out to the cohorts to get them to do the implementations, and then we can build the various demonstrators. I guess, I mean, our engagement with GA4GH, I mean, it spans all of the work streams, to be honest. I mean, I think, and we have different connections to the different work streams, I guess, through the various PIs, I mean, in the project. So, yeah.
Okay. So we just have 1 minute left for a couple of things, housekeeping things. So I think we will wrap up questions, but I would ask you to maybe answer them in the chat. In particular, I'm curious if there are any gaps in current standards for human cohorts, if you want to tackle that one. Thank you. Thank you, Thomas. Thank you, everyone, to all of our speakers today. This has been really fabulous. And Justina, if you want to go to the final slides, I think we're just going to wrap up with an overview of what's coming next for today. We have 3 working sessions this afternoon, evening, depending what time zone you are in. But starting right after this, we have a social event in spatial chat, and you can go to the next slide, Justina, but the link I think is there. We recognize that we originally had one social plan for the end, but it was like 1 a.m. Europe time and ridiculous hours. So we added a second one, and you can join us here for an hour following this session. So thank you all so much. This has been a great morning, afternoon, evening, wherever you are, and I hope to see you at all of the sessions coming up.